that.
I hope it won't get worse. That's all I hope.
All right.
Okay. I thought this article was a little trickier than the last one, so I'm going to do my best. So figure 1A shows the cycle of the 3 phases, the foraging phase followed by the consummatory phase, which is reward processes, and then satiety develops, and eventually the behavior is terminated by PBN neurons, for example, which can take you to the satiety phase. And so AGRP neurons and hypothalamic arcuate nucleus, which is the ARC, are activated by energy deficient signals and bring you back to the foraging phase. And so AGRP promote preparatory phase of hunger, but they're not required for the consummatory phase and do not have a role in sustaining food consumption. And so we wanted to see what other neuron populations do this kind of thing. And we also wanted to learn more about thirst, which is commonly controlled by neurons in the SFO. And so ultimately the goal is to use AGRP and SFO neurons as entry points to learn more about the circuits and what happens downstream of those neuron groups.
So in panel A, where would taste go in? I'm just asking a general question. Yeah. Where would taste be?
Probably between 1 and 2.
Right? Yeah.
I think this paper is also suggesting that was a homeostatic feeding is distinguished from the hedonic eating behaviors, right? It's like they want to say AGRP drives the homeostatic, so like you need energy, so you go for eating, and the rewarding system is a little bit different.
Yes, and it becomes a question of semantics. Many of the studies here, right, where hedonic versus homeostatic feeding seems like a internal state factor that affects it, but also when we talk about taste, that's also affected by the internal states and the homeostatic state of the animal and the hedonic state of the animal. I think that switches also. So I think it's not a black and white kind of a deal. In many of these the studies that they look at here, I feel like it's restricted by the semantics also, right? I'm just saying that the body states are not as simple as we would like it to be. So it's not a binary choice. Yeah.
So figure 1B shows the strategy they used of using brain sections downstream of AGRP and SFO, and they used Cree-HSV to identify them along with the TD tomato staining, and then they also did FOS mapping after the activation of the neurons during lack of food and water. And so in C, it shows that they were stained, imaged, and quantified, and sectioned. And then 1D shows the results, that there were some regions that contain an overlap of both HSV and FOS. And so the ones that the article highlighted were the PVH, LHA, and PVT, which they already knew were important and involved in these processes. So they wanted to look at brain areas that they didn't know much about. And so there was a lot of labeling, as you can see, where they focused on this one section of the hindbrain in the PCG. And in both AGRP and SFO, they found similar overlap of MEV, PCG, LC, LDT, SLF, and SCP. And so FOS-IR was found for both groups in MEVB, which I think is Barrington, and LDT. And so figure F shows that cells in dorsolateral PCG showed elevated FOS in food-deprived and water-deprived mice, but not control mice. And then figure G shows that the PVH projects to the dorsolateral PCG, and that this area is called the peri-LC, which is the focus of basically the rest of the article. And H is a view of ZI projecting to the PCG. The red axons are MEVB, PCG, LC, LDT, SLD, and then the yellow are other axon regions that were not specified. And then I and J are basically repeats of that, but with the LHA and DMH. K is an overlap of 6 additional neurons. And then, so based on all of these results, they wanted to look further in the peri-LC and its surrounding sites to look into its role in regulating food and water consumption and regulating palatability.
Very good. Amanda.
Yes.
Do you know how the HSV travels? So
no, I didn't really know the mechanism of it, but it said it used it to identify. It was like an anterograde sort of thing going
that's
on there.
Right. What does that mean, anterograde?
Future.
From where? Let's say starting from the cell bodies in the arcuate nucleus, AGRP neurons, where would that go, the virus?
Can you say that again? Sorry.
So let's say you infect the AGRP neurons in the arcuate nucleus with the HSV, which is a Cre-dependent.
Yeah.
Right?
Yeah, I just didn't really know the mechanism.
Yeah, so then from the cells that are in the AGRP expressing neurons in the arcuate nucleus, from the cell body, the virus will travel through the axons, right?
Yeah.
And then infect the cells that the axons synapse to, right? Yeah. That's what anterograde means.
Yes.
Yeah.
Okay, got it.
So cell body to axon to the next cell body to the axon to the next cell body to the axon.
Oh, got it. Okay. Is that similar to what you do with the rabies, or is that a little different?
Rabies is a retrograde one, so it will go from the axon back to the cell body and then infecting its presynaptic cells. So for anterograde tracing, you want to trace what is downstream of the region you want, so where they are going to, and the rabies is which region are projecting to this, so you're going upstream, so upstream towards.
Because they wanted to look at the cell clusters downstream, so that would make sense.
Right. They want to see a convergence.
They want to see what brain areas AGRP neurons in the arcuate nucleus would control.
Yeah. Okay. Makes sense. Thank you.
My question here is like the Cre-dependent HSV. Does it mean they use a mouse line specific expressing Cre in these neurons, or they just inject the Cre and then complement with the HSV?
Oh no, this would be the AGRP Cre animals.
Then, okay, yeah.
And then the NOS1 Cre animals.
Yes. So basically they are using the same, like very similar to what I proposed. They just make a convergence from the tracing, and then they just use the CFOS upon activation of the other region, and they find emergence.
Yeah.
Right.
And Jahal wants to do some of these, but we would be thinking of combining the AGRP and the NOS Cres and making a different driver. So it could be like AGRP Cre with NOS1 flip. So you can use the flip FRT system and the Cre-lock system at the same time, for example, in the same animal.
Is it possible to cross 2 Cre lines and to have the dual Cre expression? Is it possible to do that? Like I was thinking about like a V-glute, we have V-glute Cre and we cross with P2X2 Cre so that both neurons.
Dr. Ye would answer that.
It would be really.
So I have a bit of I think back and forth discussion or kind of argument with children. It depends. If you want to say P2X2 with V-glute2, in my limited knowledge, I don't think it's a good system for genetic ganglion.
Right.
You have to go back to check what the expression pattern for V-glute2 versus P2X2 in genetic ganglion. Most of them, most likely, they're so overlapping. Like they have a major overlapping. I don't think you either want those two Cree to have an intersect. So if you imagine like mathematically you have two sets, you want either have like a what that's Marty. So set A, and you want the sets that's common or shared by sets A and B, not by all of them. Or
so
you need to have something.
In this case, if they were to cross the HRP and the NOS1 Crees,
yeah,
I wouldn't think that there's much HRP expression in the SFO, but I don't know if the opposite is true, that NOS1 might be expressed in the arcuate nucleus also. So they might see problems that way. And Cree again it's just so powerful because you need like 4 molecules of Cree for the recombination to happen. So you don't need a high level of expression. And some of these genes are like randomly switched on at times. So then you might see expression or labeling of cells that you don't intend to. So that could be the problem with combining 2 Cree systems, Cree lines. All right. I thought figure D was kind of weird because it's not very informative, right? Figure D to figure K, D to K. I wonder if there's a better way to represent it, especially figure D. I thought was not so obvious by looking at it. But anyways, they focus on peri-LC, right? All right. Mm-hmm. So then, who's figure 2?
I got figure 2.
Okay.
Okay. So now that they funneled down to peri-LC, they wanted to see what would happen if these groups of neurons were either inhibited or activated. So first, they did a chemogenetic approach where they used SAM, or pharmacologically selective actuator modules. And basically when they injected UPSEM792, it would silence these neuron activities. So in figure A,
I'm on the wrong figure.
Oh, sorry. Yeah, that's a supplement. Okay, so for yes, figure A is just a schematic design where they transduced either one side of the peri-LC or bilaterally induced, and then transduced, and then B shows that they effectively transduce these 2 sides of the peri-LC neurons. The green here represents those that have been injected with SAM, and they wanted to show that it was not overlapping with other population neurons by using red to label the neurons that have been expressing tyrosine hydroxylase. So these groups of neurons that they targeted specifically are glutamatergic neurons. And then for C, they started injecting the UPSEM792 to inhibit these neuron populations. So for C, it is targeting, or it shows like when these 2 neuron populations have been inhibited both bilaterally and unilaterally, there was an increase in food consumption for grain pellets. And they compare that with the control group, which has been just injected with the yellow fluorescent protein. So compared to the control group, where there's no significant difference, inhibiting the peri-LC neurons led to a significant increase in food consumption. And in figure D, they did the same thing with sugar pellets. So similar results, where inhibiting led to increase in food consumption. And then for E, they did an optogenetic approach and wanted to do a place preference test. This one, it took me a while to figure it out, but basically there were two chambers, and for during the pre-testing, they wanted to see which area of the two chambers the mice were more preferred to be in. And then for the other chamber, they put lasers on it where if they entered the less preferred chamber, it would fire lasers, and that would optogenically inhibit the peri-LC neurons. And in figure E, we see that when they entered the side, which was the laser, optogenically inhibiting led to place preference in the area compared to the control group, which again has just been injected with EYFP, so there was no significant change in place conditioning. And then figure G shows that the group that have been inhibited optogenetically preferred to stay in the area where these neurons were inhibited.
So does everyone understand the experimental setup here? They would inject ARCH-T or EYFP into the peri-LC, place the fiber, right? And then there will be a laser connected to the fiber, and the mouse is placed in a box with two chambers. On one side, the laser would turn on. When the mouse enters that side, the laser would turn on. And when it goes back to the other side, then the laser would turn off. So by doing this, you could see if by inhibiting, because this is ARCH-T, by inhibiting the peri-LC neurons, whether there's some preference for the side where they get inhibition of peri-LC or the side that there's no inhibition. Yeah. And then in the next set, they do the opposite, right? They do the activation.
Yeah, so for H, they did and it shows that when these neurons were activated, there was a significant decrease in food consumption. And in figure I, they did like the opposite approach to E, where they would shine the laser when they are at the more preferred site, or where they tend to be in, which is why at the pre-stage it was significantly higher. But when these neurons were activated, they'll prefer to go to the other chamber where the laser would turn off and the neurons would not be activated.
Mm-hmm.
And then finally for J, they did like a self-photo inhibition experiment, and they noticed that there was no change in the nose pokes. So from this, they determined that these peri-LC neurons do not necessarily reinforce the specific actions, which that part kind of confused me a little bit, but the whole takeaway from this section was that inhibiting these neurons would lead to a rewarding food consumption, since in the behavior approach, they would prefer to stay in the area when these neurons were inhibited. So.
Yeah, and I think there are multiple things going on here, because even in the absence of food, there's a preference for not having ARCH or peri-LC activated, right? There's a aversion to activation of peri-LC neurons. I don't know what that means.
But I like the figure I in the test day immediately bounce back. So I guess if you have another day, probably they just return to normal.
Mm-hmm.
Anyway, this is a side thought.
Yeah.
So J actually, if you look at figure E,
Mhm.
it's the same idea. So similar idea. E shows like inactivation of peri-LC creates sort of like say, makes a mice willing to stay in the sites of when the inhibition is on, right?
Mhm.
So J is actually meaning like there are two levers. One lever is when the mice press it, it's not lever, it's nose poke.
Yeah.
So two ports, like when the mice like say put its heads into, for example, left port, if it is active, then there is a light shining, and those peri-LC neurons inhibited. So according to E, they should be more. You expect them to be more like to be inhibited, right? But this is not the case in J. So it's a different type of way of measuring. I think like
So
measuring.
what is E showing versus J?
J is operands,
Right.
learning whatever
Yeah.
way. E is also learning, but it's dealing more with the spatial perception.
But J would be more the motivation aspect, right?
Could be.
And J, in a way, is connected to the findings in D, because in D, by inhibiting peri-LC, you're getting more consumption. It does not necessarily mean that there's more motivation to eat. Is that right?
Yeah.
Yeah.
But the baseline level for sucrose intake is only half of that food palette.
Yeah.
Did it mention that the sugar palette was like significantly harder to consume,
Wow.
which could be the reason why there's less?
Mhm.
Okay.
Yeah. No, this paper, they were so meticulous about testing different aspects, but I think this is such a complex behavior in itself that we could spend an hour talking about just this one figure and what it really means, right? It's not a clear cut like more feeding, less feeding, more drinking, less drinking. It's very complicated. And when we talk about the parasubthalamic nucleus, we're probably going to have to deal with the same thing.
If we don't want to deal with it, I think the reviewers will ask that.
Right, yeah, no, we definitely will. It won't be a binary choice where, oh, there's more feeding. This is what it does, right? And the authors have done so many control experiments to try to figure out what this is, but it's not easy to register. They talk about the motor control and they did all these fancy experiments to test that. All right, let's move on. Big figure. Alex is figure 3.
Who is figure 3? No one?
I can take that one because
Okay.
it's pretty interesting. It includes so many information. I also want to try to ingest that. So figure 3 is mainly the cognitive imaging that they want to further confirm the role of these neurons in response to during the cosmometry phase. So in A, B, and C, there are the experimental setup. For A, is talking about one experimental challenge when they set up the lens.
Let me cut you off a little bit and just show you the movie. Did people watch the movie? It's pretty interesting. This is how much motion there is, right?
Pretty much the same, similar as I did.
Yes. And we're going.
I told you.
For us, this Jahao has done mini scopes and all these things. So, and we see this kind of motion in the NTS, right? So what they did is they used tungsten wires, right, to correct for this. And I'll show you the movie with the wire. See, much better. Right. So that's the real takeaway for us, that perhaps we can use these tungsten wires when our expensive microscope comes in and we want to image an area where there's a lot of motion from the mouse moving its neck. Maybe we'll have to implement these kind of technical tricks to see better.
The wire is really small. Yeah, the diameter is like around 30 microns.
Yeah.
So I think it's pretty good. You do it yourself.
So you see, it's just like a general setup. They have a lichometer set, and they have a sensor to test the leaking, and also they have a speaker. I think that is for auditory cue setup. And D, E, and F is just a general result from the three different conditions: when they're fed, when they're hungry, or they are just deprived of water. So I think here they find a very similar subgroup of neurons that will be either inhibited or activated, and also about half of them just don't respond to these.
I have a general question here.
Yeah.
So far in the previous weeks, we've seen photophotometry being used, right? Where they don't have to worry about the motion artifacts. So why do you think that they went through all this trouble to set up an imaging prep rather than just using photophotometry?
Good question.
What does that give us that we won't be able to learn from photophotometry?
What is the measure on the right side from 0 to 1 in the scale of colors? What does that mean?
AU?
Yeah.
Arbitrary unit.
Oh, okay.
Yeah.
Just made a scale, and
it's probably some measure of changes in fluorescence.
Oh God, what did I put in?
But I was looking at this and I thought, if they were to do photophotometry and they see some cells that are strongly inhibited, some cells that are strongly activated, like in D, does that mean that if you were doing photometry, there would be no change? It'll just regress to 0?
no.
Could be, right? I mean, maybe you won't see the difference, the 80 cells that are suppressed and the 80 cells that are activated by this.
Could be, but certainly you would see a dampened signal.
Yeah.
I don't think they will be cancel each other out, because the color actually, I think they normalize to itself. You cannot compare, one. I don't think you can compare the units, like say if the cell number 1 has a number of 0.8, and the other cell has maybe 0.2, also very, like say, blue in the scale bar. Because whatever we imagine image or doing the fiber photometry, we're collecting all the photons. We're kind of dealing with the kind of absolute amount of the for photons that emitted from the tissue.
Yeah.
So that's a different,
But
that's
let's
my
say,
own thoughts.
if you look at G, for example, then you have 29% that are inhibited, 25% that are activated. So worst case scenario, you would see a canceling out with photometry.
Yeah, worse now.
But of course, I'm being facetious here, and in some cases, there's too much motion artifact. We won't be able to see nice imaging, and you have to come up with these kind of engineering advances to make it work. But the five photometry would provide a nice and easy and perhaps a dirty result. But once we can see single cell level responses, we would be able to see gather much more information. It's just that there's so much challenge to getting from just measuring bulk photons to looking at it at single cell resolution. Right? Yeah. So you want to go on?
Yeah. So in H and I, what they want to talk is they want to distinguish these neurons from the AGRP neurons because they found like even they train animals to pair the auditory cue into as a food preparatory cue. So that, however, so in AGRP neurons, it is previously described that upon they received auditory cue, they should be their activity should be reduced. However, here it just says it is not going to be modulated even they receive the cue in terms of multiple trials. So their activity, either activation or inhibition, will be maintained through the confirmatory motion. So I think that's consistent with what Amanda just said in figure 1. They want to put these neurons to be corresponding to the confirmatory behavior. And in J, they further confirm that these activities remains during the consumption, right? For example, on the left, we see between the first lick and the last lick, the blue remains blue. And for cell 2, the red remains red. So they are not changed during the consumption. And in K, it's just like another figure showing that these responses will remain consistent during the lick. And in L to O, the author is trying to show a difference between the short bout and the long bout, which M and N, we can see that as expected, if we are given a longer time, either the inhibition or the activation, respectively, M and N, are the amplitude are increased. However, in O, it is a special case that it is initially increase the amplitude, but then it just drops down to to be lower than the shout bout. I think here they are trying to characterize a subgroup of neurons that will be what they said is will be tracking the consumption duration in self-placed feeding bouts, so they are able to regulate themselves. That's my understanding on what they're trying to say here. And in P and Q, they just add in the omission trial. Omission trial, I think, is also part of the behavioral training that they provide the cue, but they don't provide food. I don't know if it is, my understanding is correct. Because compared to ensure, I think omission is just like failing to provide the actual food.
Yeah, there's nothing coming out of the spout. So this is not a purely motor dependent activity.
Right. So
the act of licking based on the cue is not sufficient to stimulate or inhibit the neurons.
Yeah. So it is still related to the feeding circuits here. Yep.
Okay. Very good. And we're moving along a little bit faster than last time.
which is good because we all have things to do.
Yes. Where are we? Figure 4.
4.
And now it gets very complicated, right?
Yeah, that was extremely hard for me. I think I'm not even 100% on this, but I'll try. See what happens. So they found the 2 subpopulations of neurons, the ones that are activated and inhibited, and they're trying to figure out what's the model. So if you have more activation, more inhibition, and what's happening to which subpopulation. I have to say that E through J, it doesn't seem, it was hard for me to understand what's actually happening. So E and H are for all neurons, F and I are for inhibited neurons, and G and J are for activated neurons. And so everything on the left side is food, and everything on the right side is water. And so we have regular animals, we have food deprived animals, and we have water deprived animals. And then they try to figure out which neurons are activated in comparison to the baseline during between 0 to 24, 5 trials in food and 0 to 12 trials in water.
So why don't we start with the 4 models? What does this even mean?
Yeah. So model 1 will be a negative feedback loop, because you have the consumption behavior that will activate subpopulation 1 that will inhibit the consumption, and the consumption will inhibit subpopulation 2 that will activate more consumption, which kind of confused me. Should I go on doing this for every one of them?
Yeah. So we have time for this to percolate.
Okay. So the second model is activation of, so consumption will activate the subpopulation. We'll call them 1 and 2 because subpopulation will be hard to say every time. So consumption will activate number 1, which will activate more feeding, and that will be just the positive loop, and consumption might inhibit the subpopulation 2, which will inhibit more consumption, which is kind of, I don't know, that didn't make sense unless you want to eat more and more and more. Model number 3, so activation of 1 will inhibit feeding consumption, and will inhibit number 2. And model number 4, we have activation of the 1, and then that will activate the consumption, we have inactivation of the inhibited, and then that will inhibit the consumption. Activate consumption.
So then in the actual experiments, what they're trying to do is they're testing the neurons over time, right? Because they're going through the multiple trials.
Yes.
And seeing how the responses or the consumption during the trials affects the activity of the neurons, right?
Yes. So they're showing.
Yeah, but just if you look at the results, it seems like there's not much change over the consumption.
True. Right. The only thing they say is that, where was it? significantly correlated neurons showed gradually reduced activation or inhibition in response with successive trials. So the more trials that are being done, it's less significant. The correlation is less significant. So it's not as strong as it was at the beginning, or at least not as obvious as it was in the beginning.
Which figure shows that?
That's the thing. They say it, but I don't see it. Maybe at the end of the food deprived trials, you see that it's less significant, but it's not really, because at food deprived inhibited neurons, you can see that it's not very significant the whole way. So they say it, but I don't believe it. And probably in supplementary figures, they show it. So
but
why say it and then not show it?
If you look at K or N, right? After they reach satiety, which are the S points, then there's a significant dampening of the activity of neurons.
Yes, so here they show that the activation of the neurons in food and water deprived mice, and they show the changes in both activated and inhibitory neurons.
So then the homeostatic signals coming from the body are coming from elsewhere, and they're converging on the peri-LC neurons, but peri-LC neurons themselves don't have a mechanism to regulate itself.
Yes.
Is that what this is showing?
I think it is. It's showing that it's coming from somewhere else. So they're getting another signal from somewhere. The bottom line.
This is in spite of the fact that when you activate the peri-LC neurons, you're seeing inhibition of consumption, right?
Yes.
So why is there a disconnect there between when the animal is consuming, there's activation of peri-LC neurons, right? And some inhibition, some activation, some inhibition. But why doesn't that activation lead to inhibition of consumption?
There's more feedback from somewhere else. There's more to that behavior, I guess, than just those neurons. Maybe it also has something to do with time. So the longer you do it, then other mechanisms are activated. Either way, at the bottom line, they say that they see a stronger, more dominant effect of the inhibited population, which I don't know. I don't really understand how they got to that result.
But I think this is a cautionary tale for all of us when we consider like chemogenetic, optogenetics, and artificially manipulating activity of neurons. And where it's like hitting the neurons with a sledgehammer. And we might see things that are much stronger than what the neurons are actually programmed to do.
Well, we are a very complicated machine.
Sure.
We mice.
Should we go on or do
Yeah.
Anyone else have comments? No? And so far, they're seeing convergence of feeding and drinking, eating and drinking, but as a more of a parallel, right? They're not seeing interaction of feeding and drinking yet. Anyways, let's move on to figure 5. We're going to go over the 1 hour mark again. Figure 5. We'll go over figure 5.
So I should. What does this one mean? Honestly, I didn't look at the figure. I have to I have to remind me like what it really means.
Mm-hmm.
So I think now they're going back to another topic because they're trying to say the palatability of the food. Basically by diluting sugar, I don't know how much diluted, so make it less appealing to the food deprived animal, the food deprived. Well, they show they're using the same techniques, and with green lens imaging, you see the same population pops up with you have activated population, you have deactivated population, and if they are presented with diluted one, actually those activation or inhibition disappear, right? And then B and C is I think is another look on the like say more like a zoom in of what presented in A. And they show you that for in-beat population, the first leak of insure versus diluted insure is different, dramatically different. For diluted insure, is the response is smaller, and you got the response of core cells, and indeed at the population level is smaller, and you can also do a mean of this population, and the same basically is consistent, and C is the opposite. So still dampening of the responses for the activated population, shift the distribution towards the left, meaning you have less neurons with higher responses. And then still the same conclusion for the mean. And then they continue to try other different not other, like say, stimuli, like the water in D, sucrose in E, quinine, which is a bitter substance in F, and also these the like cross let's say comparison. So sucrose sweet, quinine bitter, citric acid, that's the sour, high salt, that's like aversion. Again, I is bitter versus sour. And now what they find,
so
basically,
go ahead.
Sorry. So basically in D to E, F, you find that compared to the insure, so that's the palatability part, the water doesn't dampen, change anything, sucrose doesn't, but quinine changes, actually, especially for those activated population. So this is probably where the idea of palatability comes in. So generally in the food deprived animal, it seems like the water also appealing to them, sucrose, yes, but not bitter. They don't want it. And G and F is another way to get across this idea. So I don't think we want to go too much of this.
Yes, but this clearly shows the advantage of being able to look at the activity of single neuron.
Yeah. You can at least, like say, track the history of neuron activity at cellular level, and you recognize that the cells in a certain in, for example, peri-LC is highly heterogeneous. It means that there are neurons being activated when the animal consuming insure, but if labor neurons may just have a decreased activity.
And so this presumably points to taste circuits modulating peri-LC activity. No? Somehow. I don't know how. And at what point?
I'm sure if I do, like, say, multi-synaptic retrograde tracing, I can go back to the genetic ganglia, and you can see the axon touching the taste buds.
Yeah. You mean from pre-LC? If you started pre-LC.
Yeah. So these pre-LC area and LC area actually are quite interesting. So the interesting part is it belongs a little bit to the medial part of the brainstem system, while the lateral part is more like a sensory. So this part actually heavily innervated, projected by, let's say, the reticular formation in the brainstem. And those reticular formation also, I think, has a specific, like, say, biased projection from NTF. I don't know exactly what the bias are, but this is a definitely interesting thing, what I am considering. I don't know.
Yeah. All right. I think we can move on. And
yeah, J to O is just another way, like similarly. So we got the idea, that's fine.
I thought it was quite interesting to look at things like F, where there's a switch, or F or G, where there's a switch from appetitive to aversive taste.
Yeah.
And there's a quick dampening of these signals.
Yeah. They're not okay. In this sense, that's very reasonable.
Yeah, and it doesn't happen with H, right? H is going from a sour and aversive taste to a high salt, which is also aversive, and you see that the signal is maintained there. The same with I, right? Yeah. So with the change in valence, whether it's appetitive or aversive, there's a quick dampening.
Yeah.
So it's probably not a direct, like, taste quality information that's being fed to a peri-LC. It's not whether is it sweet versus umami, savory.
Yeah.
That's being fed. It's more like it should this animal continue to eat or not continue to eat.
Yeah.
So you mean before here, the taste should be already pre-processed?
Yes, it's processed to a certain degree.
It's a no-go. I have another question about the clusters that we see in, what is it, D and so on.
Yeah.
How do they choose the clusters? Is it by activity or place? Am I making sense?
Yeah, I thought it was just by activity.
Yeah.
Okay.
Yeah. K means clusterity. So, like, K means, I think, is a clustering method to find the similarly, so neurons with a similar activity pattern.
Yeah.
So at the baseline, what it looks like, and then when the insure or water present, what it looks like, and then compare how similar they are and group them. Those have the most similarities into one group.
Yeah.
Okay, thanks.
All right, so they try something a little bit more intricate. Oh, not that one. Let's look at figure 6. Who's going over figure 6?
We're over, right? So I can go when I count. Just give me a help. So I don't know why the idea of they're trying to doing the leak contingent inhibition, but if you remember before they did the for example archirhodoxin, which is inhibitory opsin, and is so the inhibition happens whenever the light's on, right? And that doesn't link to the leaking exactly every time the mouse starting bite or leak. So these figures, they're trying to, I think they're trying to look closely about whether the inhibition of peri-LC actually affects the leaking moment. So at the right time when the mouse starts to leak taste the food or leak fluids, whether the inhibition of this peri-LC affected that. And this is a very, like say, it's kind of like an instance neuronal active inhibition. So in that sense, as Hu Jun mentioned before, perhaps this peri-LC neurons most likely doesn't track the taste quality, but it tracks the valence. So when the mice starting to eat, lick, and what they do is they have these apparators. And if you look at the right at the figure A, you have this capacity sensor which sends the leaking each leaks, and it sends the information to the Arduino 2 at the bottom left. And you have the Arduino 1 also, I think, is to control what Arduino 1 does. So they trying to, I think Arduino probably controls the information coming from the laser or to the laser. So you have the information of the leaking registered, and you have the information of the shutter registered, and you have the leaking control the shutter. So it means when the animal starts to leak, the shutter is on, shutter is off, and then you have the laser passing through, and then you have the peri-LC inactivated. So they try to using different computer programming to adjust the parameters so that for example, in B, you have contingent simulation where each of the green, this is not a bar, whole bar. This is like individual bar, which is stacked together. So you can have like a horizontal one. So, but it's actually I think it's an individual one, and individual one which is temporarily linked to the leaks. So each leaks follows probably a certain probability of light laser on period.
So then what they're trying to do is they're trying to see the effects of peri-LC neurons on consumption behavior. Right?
They did that before, right?
But they want to limit this, so to focus in on that part, so that if they do this immediately after licking, and you specifically inactivate peri-LC neurons as the animal is licking, what does that do? And it seems that that they're getting more feeding, right? Here, they see increased feeding and increased drinking. So then that would suggest that peri-LC is controlling consumption or the amount of consumption. Is that right? Yeah. Can we move on?
Okay.
Because next one is also kind of connected. Figure 7. Everyone's looking away, I see.
I'm looking at my bigger screen.
So I think what they want to show here is that the consumption duration when you do the locked inhibition is that they get more longer feeding, longer drinking. Which is an extension of figure 6, actually. The increase is because there's more sustained eating and drinking.
It's interesting.
What happens here? I didn't look at this carefully. The J to N. Can somebody explain J to N?
They have this is like a artificial sweetener or no sweetener. It's a yeah. It's a sweetener, but doesn't have like a caloric content, right?
Yeah.
And then you have like 2 bottles. One of them paired with the light inhibition, and supposedly they should. If we predict from what know before, it should increase the intake from the bottle that paired with the light. So K shows that. So the first bottle, actually first they start with like they don't like it, probably the bottle which they not prefer, and then inhibition leads to the preference of this bottle. Okay, goes back to, it shouldn't be treated as a chance level, right? 0.5. Yeah, it's not a chance level. They have a formula, and then if they, I may give concern about the photo inhibition second, but so since I have to look at it. A is euro 1 pretest with no. Oh. So I think the first and second is the first bottle and the second bottle. And so it means like say so they shows both the K doesn't mean like it is the time, it's not on a time scale. It's just on different conditions. And when you do stimulation of photo inhibition, you have the first bottle and second bottle consumption. Obviously, the difference is huge. While the first bottle, where the animal was not preferred, becomes like the preferred one. While the second bottle, which is previously preferred, but this time like doesn't care at all. And then after several sessions with extinction, meaning that the light doesn't pair with them. But I think there should be another cue inside with this assay, like when to indicate animal, to tell the animal when the session started, and then there's light on. So Now they're Q on, but there's no light on any of these tubes. So in this case, I think in this case, the animal doesn't, I think the learning somehow gets reversed. And I think this kind of learning, I say. AO is another way to look at this, what happens for the leaking behavior. I don't think I'm not making any sense here.
And then for M and N, what they're showing is that by inhibiting pre-LC, they can somehow get the animal to prefer pre-LC inhibition for half the amount of sweetness. And I think what makes this complicated is we don't know how that relative appetitiveness is encoded in the brain so that you can get behaviors like M and N. Right?
Sorry, I think I now got my sense back. I think I may interpret it in the wrong way. So K, the score is for first bottle. So no matter whether the light paired with the first bottle or the second bottle, the score only shows for the first bottle.
Yeah.
So it all makes sense.
Yeah.
So if they switch, so meaning that the pairing is really, I mean, the learning is reversible, and it can be extincted. So.
Yeah, I think what makes it really hard here is that this sudden shift to the taste system and the input from the taste stimulus, and we don't know where that's really coming from. So it makes it really hard to understand what this means. So when you guys next do the CFOS experiment, maybe you should look at pre-LC and what happens.
Yeah.
It's actually head up.
It's actually what?
I sent you the picture. It's activated.
Yeah, I know.
When you stimulate the P2X2, it's activated. That actually makes sense because it's part of the, I think it should be part of the, it receives part of the valence information from taste input. Okay.
So you think that from the NTS through the PBN somewhere?
It's probably not through PBN. That's a different, so probably not from PBN. My idea is from NTS, then go to some part, route some part from reticular formation.
And then up?
And then to there. They don't necessarily have to be starting from PBN.
So I guess we should be getting some HSV.
Right.
To stop sharing.
Yeah, we should wrap it up because we have like a surgery.
Yeah.
monitoring in a few minutes.
Okay. Oh, that's right. Yeah.
You forgot it.
No. I didn't realize it was 1:30 already.
Okay.
Okay. Well, I'm going to stop the recording.